Hello everyone. Today I'm going to talk about my RAP2 project, Characterization of Ventricular Volume in Junk Deficient Brains. First off, let's ask, what is junk? Junk stands for c-Jun N-terminal kinase. It is a critical signaling pathway that regulates neuronal function. The junk pathway is regulated via 3 junk genes. They're named junk 1 through 3 respectively. Loss of this function has shown to lead to disorders such as autism spectrum disorder. To better study junk, our lab used conditional genetic deletions to remove junk function from the ventral forebrain. This allows us to study its effects on the developing brain. In this model, junk 2 and 3 were knocked out of the whole mouse, while junk 1 was only knocked out of the DLX 5-6 territory. You can see this in the green. This means that only the green territory there is completely knocked out of all 3 junk genes. From previous work done in this lab, it has been shown that mice with junk triple knockout show enlarged ventricles. And then in this study, we wanted to develop a quantitative way to measure these ventricular volumes to better understand the relationship of the control brains and our triple knockout. Next, we can see the total methodology for how we turned these mice into our quantitative data. We started off by mating the mice. These mice would either express a control genotype or a triple knockout genotype. The pups were then collected at postnatal day 0, which is the day they were born. They then had to be preserved using fixation, and the mice were embedded before the brain was placed onto the scanner. Afterwards, the heads were scanned using micro CT scanner, which allowed us to view in high definition the entirety of the brain without even damaging the brain. By referencing the brain structures with the Allen Brain Atlas, as you can see in box C, the ventricles were traced by hand in order to form isolayers using the program that we use called seg3d, which, in addition, this program also allowed us to calculate the ventricular volume. These are the results we found. It was determined that not all triple knockouts had the same effect. Some were more severe than others. I've shown some models that express mild effects on the forebrain development and some that express some more severe effects. Each brain has a segmented view showing what its ventricles look like in the sagittal, coronal, and axial position. The fourth set of images, however, it's a little different. It's the rendered model of the ventricles made by the seg3d program. By viewing the images, it can easily be concluded that even mild triple knockout has more ventricular volume than the control. But the question we want to ask is by how much? To answer this question, we must take each ventricle into account and subcategorize. From the left to the right, we can see the 3D rendered models of the lateral, fourth, and third ventricles. The number of brains we had for each genotype was 3, meaning we had 3 control and 3 triple knockout. As you can see, the triple knockout lateral ventricles had a much larger volume, although the p-value is high on the triple knockout. Even the lowest point is still much higher than the control brain. The third ventricle's volume differed the most from the control counterpart. It was shown to have the greatest increase in the volume between genotypes. Finally, the fourth ventricle showed the least change between genotypes, although it did continue to go with the trend, and it showed higher volume in the conditional triple knockout. It should be noted that none of this data was shown to be statistically significant. However, this is more than likely due to the small sample that was used in this experiment. It was overall shown that the conditional triple knockout of the junk genes creates enlarged ventricles within our mice model. And then in the future, we will use this technique to measure specific parts of the mice brain in order to better determine how the function of junk affects the cortical and subcortical neuronal pathways.